So today I'm going to talk on the types of viruses, structures, and the life cycles. In this topic, we will talk about the general introduction about the viruses, their structures, some of the viruses, general structures, and the life cycles of some of the viruses. We categorize the viruses on the basis of their nucleic acid, on the basis of their, the capsulated and non-capsulated viruses, or so on. So, the first lecture we'll be discussing on the types of viruses. We have a different types of viruses. First, before we go into detail about the viruses, let me explain about the viruses. Viruses are the particles, we call them as non-living particles or living particles. So, they are at the borderline of the living and non-living, because these viruses are living when they are in the host or with the host, and these viruses are dead, are non-living when they are without the host. So, they are the host dependent entity where these viruses are dependent on the machinery of the host to survive. When there is no host, virus cannot survive. So, virus machinery, their replication, the whole cycle will dependent on the host machinery. Therefore, we call them as the borderline of the living and non-living part. Then viruses are of different kinds on the basis of their type of host also. Some of the viruses are called the plant viruses, some viruses are called the animal viruses, and some viruses are called the bacterial viruses. We call the phage viruses. Now, within the animal viruses, we have further categories. Similarly, within the plant viruses, we have further categories on the basis of the content of the nucleic acid and the type of the nucleic acid, right? And the capsomer are capsid formations, are their structure formations, their shape, and so on. Similarly, in phages also, we are categorized them into different shape, structures, and different type of the mode of cycle of these phage viruses, like lytic and lysogenic viruses. So, as per the Baltimore system of the type of viruses, following categories are defined. For example, we call them as group double-strand DNA viruses. In double-strand DNA viruses, we have the genome replications, double-strand DNA to double-stranded DNA mRNA synthesis, and then from mRNA synthesis again we form double-stranded DNA to mRNA synthesis. So, in this cycle you have a double-stranded DNA undergo replication, then replication go mRNA formations, and from mRNA formations again the proteins are formed. Whereas in the single-stranded DNA viruses, the genome replicates single-stranded DNA to double-stranded DNA, and then again to form a single-stranded DNA for the formation of viruses. And mRNA synthesis takes place from single-stranded DNA to double-stranded DNA to mRNA. So, this is the process of life cycle of the viruses, where the viruses functions in the host system. In case of double-stranded RNA viruses, in double-stranded RNA viruses, we have a genome replications of double-stranded RNA. Single-stranded RNA is formed, and then from single-stranded RNA to double-stranded RNA is formed for the replication process. Similarly, in case of mRNA synthesis, the double-stranded RNA to directly form the single-stranded mRNA. Plus strand RNA viruses and the minus strand RNA viruses, the difference between the plus strand RNA viruses and the minus strand RNA viruses is, in case of plus strand RNA viruses, the genome replicates from RNA plus to minus RNA and then plus RNA. So, finally, it is the plus RNA which replicate form the proteins, and mRNA synthesis takes place from RNA to mRNA, and then minus RNA is formed, and then mRNA is formed from the minus RNA. Then finally, RNA is converted into the plus RNA for the replication and for the assembly formations of these variant particle. Now, in case of negative RNA RNA viruses, genome replicates minus RNA to plus RNA, and then again minus RNA for finally assembly of the virion particles. For mRNA synthesis, minus RNA to form directly mRNA. In case of plus RNA, the RNA form the directly mRNA synthesis. Now, retroviruses are the category where the genome replicates single-stranded RNA to double-stranded DNA through transcriptome, through transcriptase. We call the RNA transcriptase, which form the reverse transcription. reverse transcriptase enzyme which is present in the retroviruses. Then from double-stranded DNA to single-stranded RNA is formed, and in mRNA synthesis, single-stranded RNA to form double-stranded DNA, and then mRNA is formed as usually the transcription takes place. Now, the reverse transcribing DNA viruses. In case of genome replication of reverse transcribing RNA viruses, DNA viruses, double-stranded DNA viruses, they form double-stranded DNA to single-stranded RNA, and then from single-stranded RNA to double-stranded DNA. Now, you can ask a question, why it is formed the double-stranded DNA finally, or plus RNA finally in case of RNA plus, right, for making RNA plus to RNA minus, then plus RNA. Now, this is taking place because finally at the later stages when the RNA, when the nucleic acid is assembled in the virion particles, the complete virion particles, as a new virion particles when they form, so they have the intact assembly or intact the genome of this kind of a genome. Otherwise, if the genome is not remain intact, then the viral property will be changed. So retroviruses and reverse transcribing DNA viruses share the common evolutionary history. They use the reverse transcriptase enzyme, which is present within the virus's genome in the life cycle. And nucleocytoplasmic large DNA, NCLD we call it in short, viruses have large double-stranded DNA genome and share the other characteristic features. So, this is the figure showing the slide showing the viral mRNA, the plus strand RNA, negative strand RNA, and plus strand DNA and negative stranded DNA. This is the final strands which are formed, examples of the strand which are formed from the different genome of the viruses. So the plus strand and negative strand viral genome, they are formed like this, and the genome replication immediately converted into the correct genome type of the viral particle before it get assembled as a new virus particle. Because if it doesn't take place, then the viral particle may acquire some different type of genome which is not a characteristic features of the virus particle. Now, let's have a look at the structure of these virus particle. Why I'm saying again and again virus particle? Because we call them as a virus particle as long as it is not the intact into the host system or into the interact with the host molecule or host cells. So this is the example of influenza viruses, which is very common viruses, where the spike proteins are present on the top, and these spike proteins are, see, this is the spike proteins, spike protein, and this is the RNA molecules, and this is the capsid, and this is the envelope. So influenza viruses is basically the envelope viruses. Envelope viruses are those viruses which are having the envelope on the top of the capsid. Capsid is a proteins, right? Whereas envelope is lipid membrane, which is present on the top, on the peripheral top layer of the whole the virion particles. Now, this is basically protective mechanism, but on the other hand, the viruses which are having the capsule, or which called as envelope, are basically labile viruses. They are very sensitive viruses to the heat, to the detergent, to the simple soap and so on. Whereas the viruses which are non-capsulated or non-enveloped viruses are basically the viruses which have very harsh conditions. They can tolerate the harsh conditions. They can have resistant to the temperature also. Now, this is the example of bacteriophage. Bacteriophages are the virus particle which are attacking on the bacteria. And this is the typical structure of bacteriophage, where you can see the capsid, and this is the nucleic acid. This is called head, and this is called neck, tail, and tail fibers. Now, these tail fibers are basically involved in the penetration of the phage onto the bacteria. They penetrate the bacteria through their pins, these pins. We call it tail fiber pins. All right. Now, the type of viruses here we can define with examples, genome types and the descriptions of these viruses and the examples of the viruses. Now, say double-stranded RNA virus, sorry, DNA viruses. Double-stranded DNA viruses are basically this one. Yeah, it has adenovirus. So we call adenoviruses are belong to the adenoviridae, then hepsviridae, and the herpes simplex viruses. Poxviridae is called variola viruses. So these are the example of the DNA. This is the example of the double-stranded DNA viruses. Now, single-stranded DNA viruses. In case of single-stranded DNA viruses, you have the parvoviridae and the parvovirus B19 is there. Then we have Sarcoviridae is a family where we have porcine sarconaviruses, sarcoviruses. So these are some category of the viruses which are under the category of the double-stranded, single-stranded DNA viruses. Now, when you talk about the double-stranded RNA viruses, in case of double-stranded RNA viruses, the best example is the Reoviridae. Reoviridae is carrying the rotaviruses. Rotaviruses is one of the very common viruses called, pathogenicity in the kids for the gastric imbalance and then loose motions and kind of symptoms are, and they are very deadly viruses, rotaviruses. Now, single-stranded RNA that is called plus viruses is called positive sense viruses, or positive sense single-stranded RNA viruses. Where the example is the picornaviridae. We have a polio viruses, then you have flaviviridae. In flaviviridae, we have a dengue viruses, then you have hepatitis viruses. Hepatitis C especially is a part of it. Then we have a coronaviridae, we have SARS-CoV-2, SARS-CoV-1, we call COVID-19 viruses, which is called single-stranded RNA viruses. Now, single-stranded minus strand of RNA viruses, as I have told you, there's a difference between plus and minus strand is that every minus strand virus is first to form the plus strand viruses, and then from plus strand viruses they form the again minus strand viruses. So the example is orthomyxoviridae. In orthomyxoviridae we have influenza viruses, then we have a rhabdoviridae we call the rabies virus, which we call the dog viruses, or we call viruses which is spread through the dog. And then we have a filoviridae. Filoviridae is an example of Ebola virus. Ebola virus is basically under the category of filoviridae, which is under the minus strand RNA viruses. Now we have single-stranded RNA RT virus, reverse transcriptase viruses. Now, positive sense single-stranded RNA that replicate via DNA, intermediate, we have a reverse transcriptase is present, and this reverse transcriptase is present to convert the this single-stranded RNA into the DNA, right? And this DNA when formed, then again transcribed to the mRNA formations for the protein synthesis. So this is the natural process, or the cell machinery works to replicate the viruses. An example is the HIV viruses, which is under the retroviridae, or we call the retroviruses, category of retroviruses. Then we have a double-stranded DNA RT viruses. Now, double-stranded DNA RT viruses primarily double-stranded, then converted into the reverse transcriptases into mRNA, DNA and then mRNA and then DNA. And this example is hepatitis B viruses, or this is hepadinaviridae. Hepadinaviridae is basically the category where the hepatitis B virus, which is very, very deadly viruses and highly infectious viruses. Even it is more infectious than HIV viruses, right? Now, the life cycle of these viruses, if you talk about the phage, there's two category of these phage viruses. Phage are the viruses where we have the bacteria as a host, and these phages are the viruses. Now, in the phage, we have a number of different phage particles, and these phage particles carrying different characteristic features, the shape, size, structures, and so on. Now, phage types are basically virulent phage and temperate phage. In the virulent phage, the phages are infected to the bacteria and it lysed the whole genome of the bacteria, right, to kill the bacteria, to degrade the genome of the bacteria, and they survive and they form virion particles and they're released out. Whereas in case of the lysogenic phage, the phages are in the temperate stage. Temperate stage means like phage DNA is integrated into the genome of the bacteria. So suppose this is the phage DNA, this is the bacteria. So this DNA comes, and then say for example this is the DNA phage. So this is the bacteria. Now bacteria carrying the, so this is the integration of the phage DNA. We call it temperate phage, right? Whereas in the lytic cycle, it is not like this, but it will not integrate it into the genome, but it will remain outside. Now second is the entry. How does the phage enter? So in case of phage DNA enter into the host cell, phage DNA enter into the host cells, and it is not the whole virus particle. So the virus particle remain outside. For example, this. So it is only the genome and phage, and this phage particle remain outside, right? So which is not the case of the animal or the plant viruses, where in animal and plant viruses these phages get enter into the host cell and they get replicate. So then genome fate is a viral genome remain separate, replicates independently, whereas in case of lysogenic it get integrated into the host and then prophage form is replicate along with the host DNA. The early events of the phage cycle, because phage cycle has early and late events. So in the early events, immediate transcription translation of viral genome starts and then genes form, whereas in case of the lysogenic phase, the expression of C1 repressor maintain dormancy. So C1 repressor is present where it suppress the expression of these genes for the transcription process. Therefore, the virion particles may not form, rather it will remain integrated into the genome. So this is the difference between the lysogenic and the lytic cycle. Now host cell status is host machinery hijacked, because the virus enter into the host, it hijack the host machinery and start working on it, and host cells completely destroyed. Whereas in case of lysogenic phase, host cells survive, they remain in the active stage and in continuously dividing and allowing the virus particle genome to be divided continuously in every cell. Now the phage production, many new virion are assembled. No virion productions during the lysogenic cycle, rather they remain integrated or in the dormant stage. The release of these phage particle in case of lytic and in case of lysogenic, it varies the cell size, because in case of lytic stage, the cell lyses via choline plus lysozyme, which release the 100 to 200 phage particle from a single cell. Whereas in case of lysogenic cycle, there is no lysis takes place and prophage replicate within the host DNA for as long as it remain in the dormant stage. The duration is minute to hours, very short in case of lytic cycle. In case of lysogenic cycle, it is a long and can persist for indefinitely time. Maybe it could go for years and years. Induction is not applicable in case of lytic, but induction and through stress of DNA damage can be taking place by UV light in case of lysogenic cycle, which may induce from lysogenic to lytic induction, and they undergo into the lytic induction, then release out from the cell after the virion particles are formed, and this is the killing the cells at this stage. So now the lysogenic state then convert to the lytic stage, then they kill the phages, kill the host cells, and they came out of it. The immediate death of the host, whereas in case of the viral genome pass to daughter cells until induction. So just before induction they can remain survive, but just after induction they kill the host cells also. Now, what are the different steps of a lytic cycle? The step of lytic cycle, the attachment of the host, then absorption takes place. Absorption means like this is the host and this is the, say for example, phage particle. It absorbs first. So the first thing is that attachment to the cell surface of the host is very, very important. If this attachment is blocked, then the virus particle cannot attach it. So attachment process is taken care by the virus particle as a virulence factors. We call them as highly virulent viruses which can attach. So T4 attachment initiate with the tail fibers, right? And these tail fibers can work together to form this. Then we have more tail fibers are formed. It makes like attachment machinery to interact with the phage particles and then to form the this ring, 24 rings of a long. Now, the penetration is the process where after the attachment it gets penetrate into the host cells, and this as the sheath on the surface of the virus particle, right? It get penetrate, and then after penetration it inject the nucleic acid. An injection of the nucleic acid is basically responsible for several protein factors which are governed by the host machinery or host this, and they release these enzymes we call lysozymes, which break this sheath of the surface of a sheath. Now, when the lysozymes, they break the bond linkage sugar peptidoglycan bond of the bacteria, then they pierce the nucleic acid through these pores inside the the bacterial membrane, and this allow the whole nucleic acid get penetrate inside this. Now, GPS aids the protein which aids in the penetration of the tube through the peptidoglycan layer, because peptidoglycan layer is the very tough layers which is form of NAM and NAG, as I have told you earlier in case of when we talk about the bacterial structure, ultrastructure, and this can not allow any external entity to get entered. So what these GAPs, GPS and lysozyme enzymes do, they just lyse these peptidoglycan, they dissolve it, and they allow the holes, and these are called transit pores, and they can allow the DNA to enter inside it. So this is the process of, so first is attachment, second is penetration. So without attachments and without penetrations, life cycle is not possible. Now see the example. So this is how the virulent bacteriophage, so we call them as a virulent and avirulent. So this is example of the virulent bacteriophage. Now the virulent bacteriophage means the phage which can cause the infection, and avirulent means which cannot form any kind of infection because of some avirulence status. So the first is the attachment, we call the landing, then after landing it attached to the surface, see this, and then tail is formed like this, contact to the surface here, and then penetration inside starts into the membrane, and then see the injection of the nucleic acid inside it. So this is the process of penetration and infection on the phage particle. Now look at this cycle. This is the life cycle of the bacteriophage, because bacteriophage cycle is dependent on the bacteria host, so it also depends on the bacterial cycle. Generally the bacterial cycle is go for not more than 25 minutes. So within 25 minutes the bacteria has to complete their cycle. This phage has to complete their cycle from 0 time to 22 minutes to lyse the cell. Now, because bacteria has to divide before the bacteria get divide, they just lyse the bacteria and then kill the bacteria, right? So the idea is that first injection takes place, and then these phage particles, see the phage particles are here, and these phage particles degrade the host genome cell. So this is the host genome, all the host genomes get degraded with the phage particle. Now this process is called the lytic process, which goes for 3 minutes to 4 minutes. And then these, sometime what happened, these phage particles or phage DNA can be integrated into the bacterial DNA like this. Suppose this is the phage DNA, so it get integrated into the small degraded form of these bacterial genome. Now this, when again assembled with a new virion particle before the lysis takes place, carry the bacterial genome DNA along with the phage DNA. This process we call the transduction process. And then finally go to the formation of the latest stages, the formation of the head and tail, and then from head filled with the tails are attached here, the virion form. So this is called the virion formed. Virion formed the particle which are remain in the bacteria but not released out. So before the particles are released, we call them as virion particles. Now the biosynthesis is the process. The temporal gene expression within 2 minutes after the injection of the T4 phage. And in E. coli, as one of the hosts, because it may be Klebsiella pneumonia, it may be Pseudomonas and other bacteria, but this is the typical example of E. coli where within 2 minutes after injections, T4 phage, the synthesis takes place. And E. coli RNA polymerase starts synthesizing T4 mRNA for the process of the various protein formations after the transcription takes place at the early stage of the cycle. So the early transcription within 5 minutes, where the viral DNA synthesis starts the catalyzing the virus encoded DNA dependent DNA polymerases. This DNA dependent DNA polymerase helps to form the copy of the DNA for the further replication process of the new virion particles. And in the late transcription stage, next step, which comes to be in next 4 to 7 minutes, where the viral DNA replicates and followed by the synthesis of the late mRNA. And this late mRNA, which form in the later stage of the infection, which provide the new protein formation takes place, which help in the formation of the virion particles, so that capsid, capsomeres, tail fibers, so that the virion particles are ready to proliferate or to increase in numbers. Now ADP ribosylation begins. The viral enzymes attached to the ADP ribose to form alpha subunit of a host RNA polymerase. This is the intermediate stage of the phage cycle in the bacterial host. And this effect of modifications of these takes place in the host cells, where the host gene transcription is suppressed. And so the host DNA cannot form their own replication cycle, cannot form their own growth, own proliferation, own division. No, it is stopped. So within the same bacteria, it proliferates without allowing the host to further grow. That's the key of the proliferation of this virus particle. And the second alpha subunit of RNA polymerase is also ADP ribosylated for modification, so that the E. coli or the host may not replicate or host may not propagate. Now switch in transcription is called some early gene stop being transcribed after their production, after the products are made, while middle and late viruses genes are continuously formed. Now DNA replication, protein synthesis, viral DNA is replicated later stages, and then this viral DNA is ready for the incorporation within the virion particles to form a complete virion particle. So this is how the single virion particles is formed, along with some structural proteins like a protein, capsid, tail fibers, and lysis enzymes, so that they can be ready with the fully loaded virion particles for infection to the other bacterial genome in the next cycle. So this is the maturation or assembly we call late transcription. It's 9 minutes after the infection, and then clustered genes formation takes place, and these genes for related functions for the head and tail fibers is and they get together to form So the maturation means like a virus particles which are in large number of virus particles which are formed at early and late transcription. They form several component of the virion particles, they get assembled and they form a matured virion particles. Now these matured virion particles are ready to lyse, are ready to kill the cell to released out, as we call the burst size of the virus particles, where the good number of virus particles, about 200 to 400 virus particles are released out from the cells after killing the cells. Now, then we have a concatenamers DNA processing, viral DNA synthesized in concatenamers, long linked copies, so that you can have a number of copies are formed to each virion particles. And then pacosome function means like a terminase complexes are formed, we call pacosomes, and these pacosomes generate the double-stranded DNA and use ATP hydrolysis to package slightly longer than genome length. And then they get into the DNA, into the phage heads, they get incorporated into the phage heads of the virus particles, and then circular permutations and the redundancies takes place where because packaging cuts occurs in different positions, different positions of the genome. So each virion has a terminal redundancy, circular permuted genomes. So different, for example, they can carry the sometime bacterial genome in it. So every virion particle genome may vary because of the part of the genes from the bacteria may also incorporate it as a part of a transduction process. And final assembly takes place by tail end fibers which are formed and in head and produce the mature T4 virion particle. Now the release is basically, we call the lysis, which is the last step of the virus particle, where suppose this is the bacteria and there's a lot of virus particle are formed. So after degrading the bacterial genome and stopping the replication process of the bacteria, these viral particles are formed like this. Now this number of, say for example, this is 100 to 200 virus particles are formed. Now these 100 to 200 virus particles are lysed out. Lyse means like these bacterial genome is start lysing. I just make like this. So this is for example, right. Now lysis means like they start produce lysosome enzymes, and these enzymes are killing the membrane like this, and now these virus particles are released out like this. Now these virus particles are ready to, each virus particle are ready to infect to the other bacterial populations, to the other host cells, and each virus particle, so for example 100 virus particles are released from the bacteria, and these 100 virus particles released from the bacteria, they can start ready to attack on hundreds of bacterial particles. So this is how the proliferation takes place, right? And this Hollin creates a pores in the plasma membrane, as I have told you here, 1, 2, 3, 4, 5, 6 number of pores, and then T4 lysozyme enzyme degrade the peptidoglycan layers of the cell walls. Then synergistic action takes place. Hollin allows the lysozyme enzymes to access the peptidoglycan, and then cell destruction takes place, and plasma membrane bursts out, broken down, so all the virus particles get out. So this is the process of the whole virion particles. Now after the release, each viral particles are ready to affect or to interact with the other host cells to increase the life cycles again. Now this is all about the first lectures, so we'll talk about the second lectures in a due course of time.